So that's our review of synapomorphy and homoplasy. Good morning. So here's a related review topic. Who can concisely and scientifically explain why coconuts aren't mammals? Pardon? No placenta. Okay, fine. So, yeah, coconuts apparently don't give live birth. Well, not with a placenta anyway.
Not warm-blooded?
They're not warm. Okay, so they have chloroplasts, etc. It's so the hair is it depends on how you define hair. So you've got a furry substance on the outside, but it's not hair in the homologous sense. These are analogous traits.
So I was going to say that the traits,
I stole your thunder.
That the traits of having hair or producing milk were analogous but not homologous in the way mammals do.
Right, so these are analogous traits. Indeed. So the last review point for the day. So homologies, analogies. We've got synapomorphies and homoplasies. So what I'd like to be able to get done today, we'll see, depends on how quickly we move, which I'm not forcing it, is practicing mapping traits onto a tree. So say you have a hypothesis and you want to know how certain traits evolved over time. That involves that first point, how to use trees to make guesses, educated guesses, about where certain traits evolved or were lost, trait gains and losses. That involves these terms that we've used a little bit before, ancestral traits and derived traits. We practice using those concepts and those terms. Rooting an unrooted tree, that was part of the reading assignment for today. And then also comparing parsimony versus distance, two different approaches that scientists use to build phylogenetic trees. Does anybody have any questions before we get started? At the end, if there's time, I'll discuss the assignment that was due last night, find the published phylogeny. So this is just by way of example of an issue where we have an unrooted tree. There's no clear outgroup, and there's no clear direction of the flow of time in this figure. So if I asked you present, that's all of the branch tips. Every one of these branch tips represents a currently living organism, but which one is most closely related to the common ancestor? Well, they all are. Let's map a trait onto this tree. What trait can we map onto this tree? Which one? Egg laying. Okay, so let's do egg laying. So, who's egg laying? Crocodiles and platypi. But not elephants and manatees. So this perfectly illustrates, thank you for that suggestion. I did not pay you to make that suggestion. The issue with having an unrooted tree. If we don't know where far back in time is, how would you tell me whether or not eggs were a trait that was lost in mammals, or if egg laying was a trait that was gained in these other two taxa? So without having a root on the tree, we don't have any perspective. We can't tell which direction evolution is moving in. Was egg laying an ancestral trait that was lost, or was it a trait that was gained? So when you root a tree, then you draw it from the past to the present, and it's a little bit more simple to infer the flow of time and how traits evolve. So this is practice time now to practice taking this tree and drawing it as a rooted tree. So the instructions are, you read about this, the instructions are to redraw this tree as a tree that's rooted on the branch leading to crocodiles. That is, the root of the tree should be there. So to redraw this tree, everybody work on this, paper, tablet, whatever. Redraw this tree, we've got about a minute. Where the past, where the common ancestor to all these species is there, where the root is. So for example, if I was going to start this tree, I would have the root down here, and I would have the extant phyla for example, than at the top. So what does it look like in between? Please ask questions as they arise. There should be questions. So in this case, if the root's placed there, who's the outgroup? The crocodiles, in this case, are most closely related to, not most closely related to, but they're the most directly related to the common ancestor. So they'll be an outgroup making platypi, elephants, and manatees a clade. Right, they're all going to share a common ancestor with each other. Somewhere here, from which the elephants, the manatees, and the platypi all descended. So who branches first in that group of three? So what you imagine doing is now we're moving from the root towards these species in time. So the first branch that comes off is platypi, like that. And then in the other direction from platypi, one direction is platypi, the other direction is the common ancestor of elephants and manatees. So when we root the tree on that branch leading to crocodiles, that defines that branch as the outgroup. And now we have a tree that gives us a little bit more information about how the trait of egg laying, for example, might have evolved. So what would your inference be for this particular hypothesis or tree? Was egg laying an ancestral trait that was lost, or was it the other? Right. So if crocodiles lay eggs and platypi lay eggs, the most likely explanation is that the common ancestor had egg laying and it was lost somewhere on the branch leading from the common ancestor of platypi, elephants, and manatees to the mammals. Now and I suggest you do this for practice, try redrawing the unrooted tree, putting the root somewhere else. On your own after class if you want. It's good practice though, because then when you look at the exact same phenotype and you look at the structure of the tree, you should come to a totally different conclusion about how the trait of egg laying evolved. If it was gained from if it was lost from a common ancestor, or if it was a gain somewhere on the tree. So we talk about this sort of as the polarity or the direction that evolution moves. It needs to be a rooted tree. If you don't have a rooted tree, you can't make a solid conclusion about how traits are gained or lost. Yeah.
So if you're saying that if we were to redraw it, it would give us a totally different conclusion.
So if you redrew the root, for example, on the branch leading to elephants, that would make elephants the outgroup, which would mean elephants and manatees would be more distantly related to crocodiles and platypi, and that would make it look like egg laying evolved recently.
Okay, so how do we know which one's right?
Indeed. How do you ever know which tree is right? I know you finally made it to evolution, and then we're telling you that nothing makes sense anymore and that there's no right answer. Wah, wah. Welcome to biology. So what is the approach that phylogeneticists take? is a phylogenetic tree ever truth? It's not fact, it's a hypothesis. So there is, I mean, it's kind of unsatisfying to say, well, there is no right tree. So what else have we talked about in class that helps us get a better understanding of which tree is more likely to be the tree that represents ancestry?
By using like evolutionary history and like combining a whole bunch of different things like morphology, behavior,
Sure.
genetics.
Exactly. So the more traits we look at and the more trees we draw based on more traits, we start coming to a consensus tree. One tree that keeps showing up over and over again. When we look at different traits with the same species, if all the trees are the same structure, that suggests that's the most likely representation of the true lineages. So to this point in class, we've been using trees, but we haven't made our own trees yet. We've redrawn trees, we've rotated branches around nodes to put mammals on top of a tree or squirrels on the top of the tree or the bottom of the tree or whatever. We haven't actually created our own tree based on data. So that's where we're moving next.
Could we have drawn that tree as a polytomy? With platypi, manatees, and elephants branching out from the same ancestor, or is that not possible?
So platypi, manatees, and what was the third one?
Elephants.
Elephants. And the outgroup was crocodiles. So if you want to draw, that's a route leading to crocodiles. And there's a polytomy.
But then that goes back to parsimony, right? Having two only.
So why was this tree not an option for redrawing the original unrooted tree? Right, if that original tree, what would that original tree, the unrooted tree, have to have looked like to draw a polytomy like this? Right, the original tree looked like this, unrooted tree. So it like had two sister taxa, sort of. If you want to make a polytomy from an unrooted tree, the unrooted tree would look like that. So it'd be crocodiles, platypi, for example, elephants and manatees. So you'll see that that polytomy is present in both trees. Good question. Other questions about the unrooted trees, rooting trees, the direction of evolution? So parsimony-based trees, we've already talked a bunch about the concept of parsimony. Fewest changes represents the tree that's most likely. So this is the hypothetical situation from your textbook. It's a really useful analogy, example. So we'll use it to start with. If the trait is fur color or skin color or bird plumage color or something like that, and these colors are represented on the tree, which two of the three taxa, 1, 2, and 3, are most closely related to each other? The two that share the trait, yeah? Okay, so in that case, you might resolve this polytomy by making 1 and 2 more closely related to each other than 3. So that would be a tree based on parsimony. The fewest number of changes, that is, that those two organisms share a common ancestor, and it was on this branch that, whatever color, is that brown? So brown evolved on that branch. Right there. So it's one change from the common ancestor having lighter coloration to a single change in a common ancestor of two currently existing species that both inherited the same darker brown coat color from the common ancestor. Now we have to, though, consider situations where parsimony is misleading. So parsimony is not necessarily the best way to draw trees. It's a way to estimate or build hypotheses. So we've got 3 different trees. Again, these are from your textbook. So you've seen these before. But what I've added are these blanks down at the bottom. So I want us to, again, practice assessing what sort of traits we're looking at and use the terminology. So A is the tree that I just drew. That's the parsimony tree, because it suggests there was a single evolutionary change, and that explains why 2 sister taxa share the same trait. But that's not the only way to get the pattern of phenotypes that we see at present. So I'm circling those in all 3 of the trees. So these are really the data that we have now. These are the data that these trees are built from. What these current species, outgroup 1, outgroup 2, species 1, 2, and 3 look like today. Present time, the 2 outgroup species have lighter coloration, 1 and 2 are darker. So in every one of these 3 trees, those facts are presented. The tree on the left is most likely by parsimony because it only involves 1 change. The other 2 trees, B and C, involve 2 evolutionary events. Which of those 3 trees is the correct tree? Well, first of all, there is no correct tree because they're just hypotheses. But how many different trees can you draw to explain the relationships between those 3 species? These are just a couple of examples, a few examples, 3. So if the tree in tree A is the correct structure, the correct shape, representation of species and how they're related, what is that trait? The evolution of brown that's shared between 2 sister species. Brown coat color would be, so this is not convergent evolution. So convergent evolution is what this one looks like. So when we talk about convergent evolution, and thank you for reminding me to use that term, sometimes this is also referred to as parallel evolution. That's when the same trait evolves separately twice. So 2 organisms look like they share the same trait, but it's not because they both received that trait by inheritance from a common ancestor. So maybe that's a better place to start, tree B. You've got parallel evolution. What's that kind of a trait called? Bowerbirds and sapphire rings. Right? The same trait evolved twice, but they're totally unrelated, 2 separate events. That's an analogy. And the trait term for that is homoplasy. When 2 species look like they're more closely related than they are because they share a trait. But in this case, they're not 1 and 2. The 2 species that have brown coats aren't the most closely related species, as you see in the tree. Which are the 2 most closely related species in B? 2 and 3. So that tree might be the real tree. That is, that might actually represent what happened over time as these species evolved. But the one trait that we're looking at is being misleading, because that brown coloration evolved twice separately. It's a homoplasy, the one we don't want to build trees on. What about A? Let's go back to A then. That's a synapomorphy. That's because it's a trait that was inherited by both species, 1 and 2, from a common ancestor. So that's a homologous trait. They both have it because they inherited it from that common ancestor. That's the kind of trait we want to build trees, because it's a trait that accurately represents that, yes, those species 1 and 2 really are close relatives. What about tree C? Which 2 species, if you were just looking at parsimony, you would say species 1 and 2 are most closely related because they share that brown coloration? Bless you.
Thank you.
Yeah. But what has happened? There's a loss of this trait just in the lineage leading to species 3. So they lost brown, they went back to the ancestral coat color. This is a trait or a term that we don't use as much. This is an example of a simplesiomorphy.
I have a question.
Please ask.
Is that the same as long branch attraction?
It's not quite the same concept as long branch attraction, and I'm not going to focus much on long branch attraction in the class, but that is a related topic that has to do with when you're looking at phylogenetic trees of species that are very distantly related, sometimes those long branches collapse on each other because of shared traits. So it's related, definitely. So the problem, as it says at the bottom line, parsimony is nice because it's easy to do, because it's intuitive. You look at a lot of traits between a bunch of different species and you just count up, okay, who shares similarities. The two species that share the most similarities of all the traits you're looking at must therefore be the most closely related species. The problem is that no matter how many characters or phenotypes you look at, you might never arrive at the correct relationship of all of the species for these reasons. You might be accidentally looking at some homoplasies, some plesiomorphies, and you'll never know. So let's take a look at another example. Practice time for all of you. So I've drawn, these are the relationships. These are based on genetics, these trees. So presumably these are the best representations of the relationships between these 6 species that we know. And the highlight here is that in 2 of these species, C. briggsii and C. elegans, as has been discussed, 2 of those species have XX individuals, normally females, that are hermaphrodites. So we're talking about the evolution of hermaphroditism, self-fertility. So what I'd like you to do is, using parsimony, figure out if the common ancestor in the tree was either a female or a hermaphrodite, where on this tree would changes from nematodes that have females in their populations to nematodes that have hermaphrodites in their populations, where on those branches of the tree, where would that trait change have occurred? So take a couple minutes, just try to figure out, draw a line on the branches where you think these transitions from female to hermaphrodite or from hermaphrodite to female would have happened. Best explains the data we see on the tree. And again, please ask questions if you have them as you go. So where do you start? Keep working if you want. Where do you start when you're trying to map traits, gains and losses onto a tree? How do you figure out whether or not the trait was gained or lost? So start with the tree on the left, for example. If the ancestor was a female, what's the first piece of information or data you look at on the tree to help you decide where to hypothesize that gains and losses of this trait occurred? What about the nodes?
Nodes.
Okay, so we start with a female. How many species are currently female? 4 out of 6. So we start in time with a female, and at present we have 4 out of 6 species that are female. So what do you think the overwhelming pattern on this tree is going to be? Gains of hermaphroditism or losses of femaleness? So we only have to draw really 2 gains of hermaphroditism. Where would those be? Right, so the branches, somewhere along the branches that lead to those 2 specific species, okay. So that would be a parsimony tree that would require 2 changes. You start with a female species, you end up with 4 out of 6 species that are females, so presumably the trait of femaleness for them is homologous. It's all evolved or all derived from that common ancestor. And then it only takes 2 independent evolutionary steps that transition females to hermaphrodites. Did anybody have something else that they were working on that tree? Because there are different answers. This is not the only one. I think this is the most parsimonious explanation. What about the tree on the right? What's the easiest way to get from a hermaphrodite ancestor to what we know today? 4 out of 6 species being females instead of hermaphrodites.
Is this the opposite case where it's a loss?
So how many losses do you have to get from here to here? So somebody said 4. Where would those 4 be? So example 1, I'm just going to throw this out there to start. So let's say there's a loss of hermaphroditism right there. Where else would there have to be a loss? Pardon? On the bottom, yeah, right. The branch leading to japonica would also have to be a loss of hermaphroditism because it's a female species. Okay, then what would we have to do from this point? We'd have to have a gain of hermaphroditism for C. briggsii. Okay, so that requires 3 steps. 2 losses and 1 gain. Did anybody, or has anybody, or does anybody think that they have one explanation for this tree that's more simple than that? Bless you. Could there be a single loss? There's 2 losses of hermaphroditism on this tree. Could there be one point where there's a single loss of hermaphroditism? What if we put loss of hermaphroditism there at the root? So what if the common ancestor of all of these species really was a hermaphrodite, but there are none of the descendant species that are still hermaphrodites that inherited that trait from the ancestor? So immediately after we start looking at this tree, there was a loss of hermaphroditism. How many more changes would we have to make to explain this tree? Right, so that looks like this tree. We're postulating that there was a hermaphrodite that was the ancestor, and then there was an immediate change to femaleness, and that's basically then the start of this tree. So that would require loss of hermaphroditism at the root plus 2 gains of hermaphroditism, which is also 3 changes. So it's as likely an explanation by parsimony. So if anybody, I'm going to leave this be now, move on to something else, unless there are questions, but see if you can come up with an explanation that's more parsimonious. Is there any way to get either of these trees from the ancestor to what we see currently in the extant species using 2 changes? That would be a more parsimonious explanation. Questions or concerns? This is an example of how intuitive parsimony is, yet how it's not always necessarily helpful in building trees. If this was a tree truly based on parsimony, we would make C. elegans and C. briggsii sister species if we were just looking at the trait of hermaphroditism, because they share that trait. But because these are the genetic trees, which we are pretty sure are the most reliable, we know that those are two very distantly related species. They just happen to have separately evolved the same trait. Parsimony, sometimes misleading. Distance, more often used. So here we get to do a little bit of practice building distance trees. And that's the last topic for the day. So the concept of distance is almost as straightforward as parsimony. Normally, distance trees are built on genes, DNA sequences. So you take the same DNA sequence from multiple different species, you line them all up like they have here. So a critical point these should be homologous sequences if we're going to build accurate trees with them. So they need to be the same part of the genome from two different species. The same gene, that is, from two different species. So in this case, you take species A and B, you line up their DNA sequences so that you're looking at homologous nucleotides. And then you simply count how many changes are there. That gives you a measure of how genetically distant two species are from each other. The more DNA changes that occur in their genes, the more distantly related they are. Pretty straightforward. And in this case, I make the point that when they say genetic distance on this chart, they're not talking about map units or centimorgans. They're talking about numbers of nucleotide changes between two different genes, or the same gene in two different species.
That could also be like amino acid changes.
It could be, yeah. So we could definitely have a very long and involved conversation about whether or not it's better to use nucleotide sequence changes or amino acid sequence changes. And that gets at one of the cons. One of the reasons not to use this sort of a distance method is, A, it's pro, it's really easy to do. So we're about to do it by hand. One of the problems is it doesn't, as it says up there, doesn't incorporate models of how traits evolve. What that means is, you may remember from biochemistry, and you might not remember from biochemistry. Are some mutations more common than others? Like what sorts of mutations are most common?
The SNPs, yeah.
So single nucleotide changes. So that's kind of what we're looking at here. So here's an example of a single nucleotide polymorphism. It's a mutation, a chemical mutation usually, of one nucleotide to another nucleotide. Now this is an unrooted tree. That is, we don't know which one of these species represents the ancestral. So we don't know if there was a mutation of an A to a G or a G to an A. But that's one type of mutation. One nucleotide chemically changes to be a different nucleotide. But there are some of those mutations that are more likely to occur than others. So transitions and transversions have different frequencies. You're more likely to see maybe A's turn into T's than A's to turn into G's. And simple distance methods don't account for that. We're just counting numbers of changes. We don't have any information in here about how likely any of these changes are to occur. So in other words, this G to A change, SNP1, single nucleotide polymorphism 1, is counted, weighted exactly the same as the second mutation, A to C or C to A. So we just count that as two differences between those two genes. But if an A to C mutation is much more rare, maybe that character trait, phenotype, should be counted more heavily in assessing the relationships between the species. We don't do that in simple distance trees. That's what we're going to do next. So we're ignoring all the complicated mess that we could get into maybe advanced evolution or population genetics classes. So, to you, here's the same DNA sequence. I say this is a homologous gene, sequence of a homologous gene from 4 different species. Which 2 are the most closely related species? Which 2 share the most nucleotides in common? So, okay, I heard somebody whisper the first and the second. Does anybody else have a different thought? Which 2 are the most closely related in terms of numbers of nucleotides shared together? 1 and 3. So that has 2 shared nucleotides. The first A and the first T. Species 1 and 2 have how many shared? 3. So they must be more closely related or more distantly related species? More closely related. They share more characters, they share more traits, they're more like sister species. Okay. So that's the essence of building a distance tree. Take a lot of homologous DNA sequences from a bunch of different species, however many species you're interested in studying. You line up the DNA sequence like this and you just count how many changes in every pairwise comparison. So we'd also have to look at species 1 to species 4 and tally up how many similarities are there. Then we'd have to do 2 and 4 and 3 and 4. So to calculate a distance tree for these 4 species, we would have to do 5 different pairwise comparisons. How close are 1 and 2, 1 and 3, 1 and 4, 2 and 3, 2 and 4, and 3 and 4. So did I miss one? I think. 2 and 4. No, I got that one. Okay, so here's practice. Let's start with practice number 1. We've got 4 species, 5 nucleotides in each gene. I've drawn a little matrix here, which you'll see show up on exams. So first question, don't put the styluses down, styli, just a second. Let's do the simple thing first. What's the distance, pairwise distance between species A and species A?
0.
There are 0 differences. In this case, we're going to tally, it doesn't matter if you tally similarities or differences. Let's count how many are similar. Distance, yeah, I'll agree with you. Let's do 0. So there's 0 differences. So there's 0 distance apart. What about B and B? Species B and species B have the same sequence. So the diagonal on these are always going to be 0. Each species DNA is the same as that same species DNA. Okay. So now you're left with, you don't have to fill in the bottom half because it's exactly the same as, the lower left is exactly the same as the top right. So now that we're there, go ahead and fill out this chart. So how many differences are there between A and B, A and C, A and D in both of these examples? most important question. Did I screw up?
My question is, do I understand this?
Pardon?
My question is, do I understand this?
So, how many nucleotide differences are there in the top example between sequence A and sequence B? So these A's are the same, T's are the same. There's a change, that's one. There's a change, that's the second change. So this distance, the sequence distance between sequence A and sequence B is 2 nucleotides different.
Because B does not have C and G?
Correct. So there's a difference there. There's a difference there between A and B. So there are 2 sequence differences, so their distance is 2. Other questions? Because, yeah, this is the essence.
So it doesn't necessarily matter what order they're in?
The order is critical, so you have to look down a column. So think about it this way, perhaps. Each column represents one position in the sequence of a gene. So if these are the same gene, then the sequence should be the same in all these organisms. There's one change. So in the common ancestor, what's the most likely nucleotide that was at that spot in that gene? For this column of nucleotides, an A. Yeah, so that's one sequence change between that species and each of those 3. And then you move on to the next column. So that's a different position in the sequence of the same gene from 4 different species. They all share a T. There's 0 distance in that trait. So if we're, for example, calculating the distance between those 2 species, A and D, there would be one change there because there's an A to C change. Where else are there differences between species A and D? There's a second one there. There's a third change there, and there's a fourth change there. So that's why I wrote there was 4 changes when I did the pairwise comparison of the sequences from species A and D. So that's the process of doing the distance calculations. One column asks how many changes per column between the 2 species you're looking at. You do every pairwise comparison of each, in this case, of 4 species. And we'll have more practice on this next class as well. But just to fill this out so you see where we're headed, then we can back up and do more examples if you want. For the data at the top, for this distance matrix, it's called, once it's filled out, which are the most closely related species by sequence? So A and B have 2 differences. So they're the most closely related sequences. So they're going to look like sister species. They're the most, 2 of the 4, they're the closest related. Then who's most next closely related to A and B? So C has 3 changes from A and 3 changes from B. So we'll draw C somewhere else in the tree, right? You could always start a tree like this, because this is always the first structure that you'll draw in any tree. 2 sister species and an outgroup. We could even put branch lengths on these trees. So the genetic distance in nucleotides change from A to B was 2. So a good way to start drawing these trees is to assume that each of those branch lengths is 1 nucleotide change. So you can actually relate the physical distances of the branches you're drawing to the number of nucleotide changes between the species. So if that's the case, then what's the distance of the branch from the red branch I drew here, from the common ancestor of A and B to C? Right, so the total from A to C has to be 3, and the total from B to C has to be 3. So we might make that branch a length 2 for now. We might have to change it, we're going to see. So hopefully so far this makes sense. So the distance from branch lengths from A to B is 2, the sum of those two branches. That's the same as we saw in this table. And from A to C, the length is 3. From B to C, the length of the branches is 3. So then what's the last question? This is the tricky part. Yeah, where do you put D so that it's total branch length from A to D and B to D is 4, but from C to D it's 3. So I'm going to leave this with you to think about for next class. Don't head off just yet. I've got 1 minute maybe. I want to run through what to do for next class. So this is the take-home project. Where do you draw D on this tree, and what's the length of the branch that leads to it to satisfy all of the conditions in that top matrix? Okay. So quickly, even though you hate having to see me at 8 in the morning, I'm hoping that some of you are actually interested in evolution and maybe you want to do a master's degree, especially if you don't know what you want to do in life, but you know you want to get some experience doing research. This afternoon in the Satellite Student Union, across the street, there's a grad studies resource fair where the biotech master's program and the biology program from here are going to be represented. We'll have a table. We'll be talking about how to get into the programs, the benefits of the programs. If you're interested in grad school here in biology or biotechnology, please stop by. I have some openings in my research group, so I'm interested in talking to people as well. You probably saw on Blackboard that I posted the details of picking a topic for your term paper. So if you haven't seen those yet, please read over them. We'll talk about them a little bit more in detail next class, as well as go over some of your results from the assignment that was due yesterday, starting to work on getting the proper formats for references for the term paper. Okay. So don't worry about Socrative. No reading assignment. So we're going to keep talking. Because we need to do more practice in phylogenetics, we're going to push back the next topic that's on the syllabus. So don't read anything, but work on that tree problem. Come back with a solution.